Now we are coming to what are the different type of culture media are present. We have to see. The first one is MS nutrient medium. Now what is the meaning of MS? First letter of two scientists who joined together prepared this medium. The first scientist is Murashik and the second scientist is Skoog. So Murashik and Skoog joined together, prepared this culture medium. So we are calling this as MS culture medium, nutrient medium. It is the source of carbon, vitamins and the hormones. So the MS medium, large quantity or sufficient quantity of carbon, vitamins and the hormones will be present. The second culture medium is B5 medium. This B5 medium was prepared by Gamboge, B-O-G, B-O-R-G will be there, but we have to pronounce as Gamboge. The next medium is white medium. It was developed by White. The last one is niche medium. The niche medium was prepared by Niche and Niche, two scientists, same name. So niche and niche. So niche medium. All these media are supporting the tissue culture technique. Now we are coming to culture conditions. So okay, the culture research is going on. The development of the tissue culture is going on. Now what are the conditions we have to maintain in that particular place, whether it is a culture medium or the culture room, wherever it is. Now the pH should be 5.6 to 6. So the culture medium, it can be liquid or solid. Now the pH of the culture medium should be ranging from 5.6 to 6. Okay, the next one is optimum temperature. What is that optimum? Very, very suitable temperature. The optimum temperature is 25 degrees C, plus or minus 2 degrees C. It can be minus 2 or it can be plus 2. So in that range only the temperature should not be there. It should not be below 23 and it should not be above 27. So that is 25 plus or minus 2 degrees C. Humidity, that means amount of water vapor present in that area. The humidity is 50 to 60%, and that humidity, the same level should be there for 16 hours. Then illumination means light intensity. The illumination, the light should be cool white fluorescent light. Now in the fluorescent light, if you take different colors will be there, green, orange, yellow, like that many things will be there. But it should be white fluorescent light with the intensity of 1000 lux. The last one is aeration. The entire research cabin should be well aerated, but microorganisms should not be present. So for that, how to aerate it, we have to see that. Now the shaking the flask, suppose the culture medium is there in a conical flask, the culture medium is present in the beaker, or the culture medium is present in the test tube. It should be shaken very well, then only the air will mix with it. Now how it should be shaken? By automatic shaker, one machine is there. Now you can fill that culture medium in a beaker or in a vessel and you have to keep it in that and it will shake so that air will proportionately mix with that. Automatic shaker or aeration by passing the filtered sterilized air. Otherwise the air should be entering into the culture medium, liquid culture medium. That is called air can be passed and filtered and sterilized air. Okay, now we are coming to the now everything is ready. Now we have to take how the induction of callus, how the callus is developed, we have to see. Now this is the explant, one small leaf what is taken from the plant. Now the explant is sterilized very well. Now that means it is washed with the tap water, then mercury chloride, so many things. It is completely cleared. Now it is not having any microorganism. So from the leaf, we have to take a group of cells. Okay, the group of cells we can call as what? Tissue. Now once you isolate the group of cells, it is also called the inoculum. The group of cells we can call as inoculum, tissue or inoculum. That inoculum, which is the group of cells which is isolated, should be transferred into a beaker containing culture medium. Now see that culture medium, MS medium. First of basically we have to take MS medium plus one plant hormone called auxin. Proportionately it is mixed up there. So MS medium with auxin. Now that's all. Now we have to maintain the temperature, light intensity, everything should be normal. Then the cells present in the group, maybe 10 or 15 cells will be there. They keep on growing and multiplying because the culture medium is supplying food. So the cells are absorbing the food. They have suitable temperature, they have suitable light. the oxygen is inducing, then the cells are keep on multiplying and it is becoming a large mass of cells, very big mass of cells, maybe 10 times, 15 times it is increased in size. Now this grown up mass of cells we can call as callus. So from the group of cells taken from the leaf, one small group, we are preparing one big callus. Okay, so like that any number of tissues we can take, any number of inoculum we can take from the leaf, maybe 20, 30, 40 inoculum we can take, separately we can keep. So how many inoculum we are taking from single leaf, that many number of callus will be formed. The next one is embryogenesis. Now the callus, a mass of cells we have studied, we have seen. Now we can see by naked eye, the mass of cells, the callus we can see by naked eye. Now what we have to do is, we have to cut the callus into small pieces. Maybe 10 pieces to 15 pieces, like that we can cut. So one callus can be cut into, imagine the 10 pieces. Then we are providing culture medium, we are providing hormones, everything. Then each piece of the callus will develop into one embryoid. Embryoid means embryo. Embryoid. Suppose 10 pieces we are cutting from the callus, then 10 embryos will be produced. The 10 embryos only we are calling it as embryoids. Then each embryo is developing into one plantlet. Plantlet means a tiny plant. So here 10 embryoids are there, then 10 plants can be prepared, can be developed. So one callus, minimum if you take 10 plantlets will be produced, young plants will be produced. Suppose imagine from that one leaf, about 15, 20, there is a inoculum we are taking, 20 callus are produced. Each callus is 10 plantlet, 20 into 10, 200 plants can be prepared from single leaf. What I am telling is minimum. So that is actually called the embryogenesis. Genesis means formation, embryo means embryo. Genesis, formation of embryo. Callus is developing into, the pieces of callus develop into embryoids. The embryoids are developing into plantlets. Now hardening. Okay, the tiny plantlets are produced in the test tube or beaker. But we cannot keep them, we cannot make them to grow big plant in the laboratory. Then we have to make it as a hardening. What is the meaning of a hardening? Just first I'll explain that, then you can understand what is the meaning of hardening. Gradual exposure of plantlets to the natural environmental condition. What is the natural environmental condition? Garden. Garden is a natural environmental garden. Plant nursery we can say. Or the field where we are growing the culturing the plant, that is called a natural environmental condition. Suppose from the laboratory plantlets are there, all in a sudden you cannot take it to the garden and you cannot keep it, because the natural climatic condition will affect, so all the plantlets will die. But what we have to do is, gradually we have to expose them, little by little we have to expose, so that the plantlets will gradually adjust with the natural condition. Because so far they are in the artificial condition. Now they are adjusting with the natural environmental condition. This gradual exposure of the plantlets from the lab condition to natural environmental condition is called hardening. Now I am taking this plantlets. Okay, they are all present in the laboratory, research laboratory. They have grown very well. Then we have to transfer them to greenhouse. So where the research is going on, there one outer one place will be there, where the glass house will be there. That is called a greenhouse. So that it is an intermediate between the lab as well as the natural condition. So first we have to keep the plantlets in the greenhouse for about 1 week or 10 days. Then they will be adjusting with the greenhouse. From here the plant can be taken to the garden, natural environmental garden. There is a condition, nursery or garden. Then the plants can successfully adjust with this condition. This is gradual. This is only called a gradual.